What does beauty mean in science, and why does it matter to scientists? These questions drove the opening plenary session of the International Symposium on Beauty at Work that we held in Washington, D.C. in May 2023. We're going to share this discussion with you over the next couple of podcast episodes. In this first episode, we're going to hear opening remarks from Dr. Peter Kilpatrick, President of the Catholic University of America, and then we're going to hear a discussion between four scientists: Dr. Robert Gilbert, Professor of Biochemistry at the University of Oxford, Dr. Dominguez, Professor of Physics and Provost of the Catholic University of America, Dr. Massimo Robberto of the Space Telescope Science Institute, and Dr. Teresa Landi of the National Cancer Institute at NIH, who moderated the conversation. There's some beautiful visuals and videos that you can also watch on our YouTube channel, which is in the link in the show notes. The symposium was sponsored by Templeton Religion Trust, the Institute for Advanced Catholic Studies at the University of Southern California, the De Nicola Center for Ethics and Culture at the University of Notre Dame, the Archbridge Institute, and the Institute for Human Ecology at the Catholic University of America. We now start with President Kilpatrick's remarks.
Thank you, Brandon. I want to add my welcome to everyone here at the symposium. It sounds like an exciting interdisciplinary venture. And I shared with Brandon that I've actually lectured on beauty and science. So I thought I would just share a few reflections, just what I've learned about beauty in my own investigation. So we typically characterize persons or created things as beautiful as being awesome or majestic, wonderful, mysterious, full of splendor, thrilling, or humbling. A dictionary definition of beauty is a pleasing quality associated with harmony of form or color, excellence of craftsmanship, truthfulness, originality, or other often unspecifiable property. Aquinas asserted that beauty has three key elements, which he called integritas, consonantia, and claritas. Integritas is the Latin word which roughly translates one or whole. So something about beauty must be whole or integral. Consonantia roughly translates as agreement or concord and is equivalent in some respects to harmony, symmetry, and proportion. And claritas roughly translates as brightness, clarity, or radiance. So one can point to many aspects of classical and man-made beauty in which a premium is placed on symmetry, harmony, proportion, and much has been written about this. Much classical art, classical architecture, natural forms revolve around the elegance of proportion. For example, implicit in the golden rectangle, or the irrational number phi, or the Fibonacci sequence. In nature, symmetry and proportion are so commonplace that an entire field of both mathematics and science has arisen around this called group theory. Many of the most important discoveries in science have occurred because of the existence of symmetry, simplicity, elegance, and even supersymmetry, as one descends to the smallest of size scales and the most primitive of bits of matter, quarks, leptons, and the like. Murray Gell-Mann, the Nobel Prize-winning physicist, who arguably did the most to invent the standard model of subatomic particle physics, famously wrote in 1969 that, quote, "One thing that makes the adventure of working in our field particularly rewarding, especially in attempting to improve the theory, is that a chief criterion for the selection of a correct scientific hypothesis seems to be the criterion of beauty and simplicity, or equivalently elegance. And yet, there is important strangeness or uniqueness about the elegance and simplicity of the laws of physics. For example, the universe is unexplainably two-dimensional and low entropy. The physical constants that define the ways the laws of physics unfold appear to be so finely tuned that our own universe appears to be designed with specific ends in mind. Theorists posit that the only way this can be is that there must be an infinity of universes, and we must be in that very finely tuned one that allows for sentient life to exist. All of this points to the uniqueness and particularity of our own created world. And this, I would assert, is the other critically important dimension of beauty, namely uniqueness, mystery, or what Aquinas called claritas, or what the social political philosopher Jacques Maritain calls radiance or radiant form. One explanation for this is that all life, all matter, is a one-of-a-kind individual incarnation of matter because of the infinite variety of ways in which matter can be constructed. A theist would assert that this uniqueness is a result of all instantiations of matter flowing from the mind of a creator who values each individual creation as the providential beneficiary of God's immense overflowing love and creativity. Thank you for the opportunity to share these thoughts with you, and best wishes for a successful symposium.
Thank you, President Kilpatrick. I now want to introduce our moderator for this evening's panel on beauty in science and why it matters. Dr. Maria Teresa Landi received her MD summa cum laude from the University of Milan and was trained in oncology and general medicine at the San Raffaele Hospital in Milan. She received her PhD in occupational medicine and industrial hygiene, and then in 2006 she received tenure in the National Cancer Institute's Division of Cancer Epidemiology and Genetics. And in 2021 was appointed Senior Advisor for Genomic Epidemiology in their Transdivisional Research Program. Her research focuses on understanding tumor etiology and evolution for potential translational applications. She leads large-scale multidisciplinary research projects primarily on genetic and environmental determinants of lung cancer and melanoma. Dr. Landi, thank you for joining us and for moderating this evening's panel. And panelists, you can take the stage.
Thank you, Brandon, for the opportunity. Thank you for your kind words. So I have really the great honor to introduce here this distinct panelist. So I will begin with Robert. I have a little bio. So Professor Robert Gilbert has been tutor in biochemistry and a fellow at Magdalen College in Oxford since 2002. He has been professor of biophysics in the Nuffield Department of Medicine at Oxford since 2015 and director of the Medical Sciences Graduate School at Oxford since 2017. Notably, he was ordained a priest in the Church of England in 2011 and has served as an associate priest in 4 parishes in Oxford and currently in Haddington Quarry, the Church of C.S. Lewis, and New Marston. Rob is also the author of Science and the Truthfulness of Beauty from 2017. And his own academic specialty is in structural biology and its application to an improved understanding of informational flow in the cell signaling, and virology. Second, Dr. Aaron Dominguez is the provost and ordinary professor of physics here at the Catholic University of America. His main area of research is in using particle colliders to search for new physics, including the recently discovered Higgs boson. His area of expertise is in instrumentation, designing, building, and using silicon-charged particle trackers as precision tools to reconstruct the complicated interactions taking place in these collisions. He received his undergraduate degrees from Whitman College and Caltech, and his PhD in physics from UC San Diego, and was a postdoctoral doctoral researcher at Lawrence Berkeley National Laboratory. In 2005, he was the recipient of the NNS Career Award. and he has played numerous leadership positions in particle physics experiments at the CERN laboratory in Switzerland and the Fermilab in Chicago. As the provost, Dr. Dominguez is the chief academic officer of the university and oversees the academic life of Catholic University's 12 schools, its libraries, research institutes, and the Catholic University of America Press. He aims to change the course of students' lives and propel them toward a meaningful, I would say beautiful, future. And then we have the last but not least, Dr. Massimo Robberto is an observatory scientist at the Space Telescope Science Institute in Baltimore, where he leads the team of astrophysicists assigned to NIRCam, the main instrument of the James Webb Space Telescope, very well known now. He's also a research scientist at the Center for Astrophysics of the Johns Hopkins University. He studied at the University of Turin in Italy, where he obtained a summa cum laude degree in physics and a PhD in astronomy. In his career, he worked as a research astronomer at the Astrophysical Observatory of Turin and as a staff astronomer at Max Planck Institute for Astronomy in Heidelberg, before being selected by the European Space Agency to work on the Hubble Space Telescope in Baltimore. His main research field is star and planet formation. He has led multiple large Hubble programs, including 2 Treasury programs of the Orion Nebula cluster that have produced some of the most beautiful images ever made by the Hubble telescope. And very important, he also has an asteroid with this name. Asteroid Robberto is 1 kilometer rock orbiting at 270 million miles from the sun. So welcome the 3 speakers here. So to begin the conversation, I would like to ask each one of you to describe an experience of beauty that you had in your scientific research. So a moment in which studying something or a discovery, you say, that's beautiful. And tell us why. If you have some, I know some of you have slides, right? Okay, so what is the, here? Yes.
This is my slide, and it's a slide which is both contemporary and historical. So on the bottom right, you can see the molecule EDTA, which chemists will be very familiar with, and biochemists. And EDTA is a molecule which binds divalent cations. It's something you learn about in your final years at school. It's something I learned about in my final year or 2 at school. And I thought at the time, when I was 17 or 18, I thought, how beautiful. I thought, how wonderful that we can have a sense of the way in which this organic molecule encases this divalent, this metal cation, and the ways in which it's curled around and its shape is suited to engaging with that cation and bonding with it electrostatically. So that was quite an early experience, and it's in fact one of the kinds of formational experiences which drove me towards reading biochemistry at university and becoming a research biochemist and a structural biologist. And thinking back to what I saw of beauty in that, it's to do with the form of the thing, and it's also to do with the way that the form tells me something about how it works. That there's this shapeliness to the interaction, they are well suited to each other, this positively charged metal ion and this organic molecule that's encasing it. And there's a beautiful shape that I thought it formed. Indeed, I went home and I made myself a model. I had a modeling kit. I made myself a model of this molecule because I thought it was beautiful when I studied it at school. So that's reaching back to when I was 17 or 18. And the main image on the screen is actually from a paper that I submitted last night. And this is from some of our current work. And this, to me, is very beautiful. And if I was to say why there's beauty in these images, I would point, first of all, to a kind of beauty that I think you, hopefully, would all be able to agree with me as well, and my 17 or 18-year-old self would be able to see as well, which is that there is a formliness to it. Up the middle there, you have this yellow ribbon of a piece of RNA, which has a very specific fold. This is actually a microRNA, or pre-microRNA. It's a piece of RNA which is involved in master regulating cell fate. And in fact, if this microRNA doesn't get matured into a mature microRNA that will engage in what is called RNA interference, as a master switch in sulfate, then someone will be more susceptible to cancer. It's a very important tumor suppressor molecule, this yellow piece of RNA. But you can see it has a certain form to it. It has a double helical structure, as double-stranded pieces of RNA and DNA do. So there's a certain river-like helical shapeliness to it, and a beauty, I think, which is perhaps discernible whether you're a specialist or not. And then behind it, there's a protein molecule to which it is suited, it's fitted. So just like the EDTA molecule is well suited to binding a positively charged metal ion, this protein here is well suited to binding this piece of RNA. And the untutored mind can see a sort of form and a suitability of these two structures to each other, which is satisfying. But for me as a biochemist, I see that. But what I also see is that I can delve in more closely and understand that fittingness in very precise detail. And so I've just got a small window at the top right there showing how the piece of RNA in yellow is interacting with the protein in a very precise way, and that helps us to understand how this recognition is going on and what are the precise atomic recognition events between particular pieces of the RNA and pieces of the protein that are bringing about this binding. All of this is very subjective, and this subjectivity is a lot of what gives scientists like me pleasure, and it's a lot of what actually drives a lot of us to do what we do. In fact, probably all of us to do what we do when we do structural biology. We delight in what it shows us of how the world works. And my colleagues will talk incessantly about the beauty of their results. And they will cry out in a satisfied way where they see the elegance, the fit, the simplicity, the ways in which you get an answer, the clarity that you obtain by seeing how one thing fits another. But what is remarkable for me is that not only is there what I think is a beauty there, that fittingness, that form, that coherence that you see as one molecule engages with another, but there's also, from that subjectivity and from that subjective engagement and that pleasure, there's a capacity to really understand in detail how something is working. And in this case, potentially in the future, to do something about the cancers that happen when this RNA gets mistreated by this protein, gets modified in a way that leads to dysregulation of sulfate and the causing of a whole range of cancers this RNA is a master switch against. And that kind of insight is shown on the top right, that detailed chemical insight. And that, for me, is one of the most striking things about the beauty scientists feel in my field. There's something subjective and pleasurable about the engagement we have with what we study, and that is, frankly, why we do it, because we find it beautiful. But the fact is that subjectivity gives us objective understanding, and that objective understanding, in the right hands, helps us design drugs that stop people dying. And there's nothing at all subjective about that. I suspect I'm going to have time to talk a little bit more about that point in a few minutes, but I just want to make that point now, that here you have a way in which the subjective leads into the objective precisely because of the way in which we engage with the beauty of something and value it for itself.
Great. So, in a way, the beginning, I want to go into the beginning of your description when you spoke about the form and that struck you from the beginning, from when you were 17, and you are a structural biologist. Yes. So would you say that in your field as a structural biologist, so form is one of the major features,
Absolutely.
Linked to beauty?
So what we're seeking is a three-dimensional description of the chemistry of biological molecules. So we're looking at the form of the chemistry, the three-dimensional space in which the chemistry happens. And as it happens, it is the case that three-dimensional chemistry is carried out by protein molecules and things like nucleic acids like DNA and RNA, which everybody now knows about because of vaccines.
Yes.
So the point is that DNA and RNA, proteins, whatever, these molecules interact with each other in a way which has a form, a very defined shape.
Right.
And that shape is built on the chemistry of those molecules, and it tells us how they work. But there's a real delight in seeing the engagement,
of each part with the others.
Yes. Yeah. In the same way, I don't think it's very different, frankly, to the way in which a child is pleased that a Lego brick fits on another Lego brick, or the way in which a block fits in a shape of the right kind if they're a very small child.
Right.
I think there's the same kind of satisfaction. It's just as adults we're playing with kind of rather more sophisticated things, and we're discovering that the world is fitted to itself, just like we can fit things to themselves as children when we play.
Yeah, thank you. Aaron, what do you think? Can I proceed with your, yes?
I've got a couple of slides here. So this is an image, it's a composite photograph of the experiment that my collaborators and I built in Geneva, Switzerland, called the compact muon solenoid. And you can't really tell from the scale of the photograph because there's not a person in there, but it's huge. It's as large as like a 5-story building. So it would fit like, not quite fit in the basilica right across the street. It's huge. It's a gigantic thing. It's buried underground, 150 meters underground in France, just across the border from Switzerland. And the Large Hadron Collider's beam pipe goes right through the very heart of this detector. And I wanted to show this image, and then what comes next is the output of this experiment, a visual representation of what we see. But just the detector itself, I think, is beautiful. I think there's beauty in the way that we've designed it, and it comes from you see natural symmetries in the way that we've built the experiment, right? You see patterns. You see kind of a harmony of pieces that we've put together, and we've designed it with a specific purpose in mind, which is to study what happens when these particle beams collide, and knowing in advance the kind of physics that we've already discovered over the past 100 or so years. We saw, like in the video that Brandon showed, Maxwell's equations. That helps actually us to design this in the right way to allow us to see the results of these particle collisions. And so you see built into our human designed machine the built-in symmetries of the universe, right? The built-in beauty of the universe that we're responding to with a machine in order to better study the beauty of this universe. So let's go to the next slide. So here's the results of one of these collisions of the proton beams. So the proton beams are accelerated to pretty close to the speed of light, about 14 trillion electron volts of energy. And these bunches of protons collide 40 million times a second in the middle of that detector that you saw on the previous slide, and create a little miniature nuclear explosion with the average energies present in that collision were about the average energies present in the early universe, a trillionth of a second after the inception of the universe, a trillionth of a second after the Big Bang. We reproduce the types of energies and interactions that were taking place in the very earliest stages of the universe. So what we try to do as particle physicists is understand what are these basic building blocks of nature, what are the rules for putting them together, how does it all work? And this image here, if there's a timestamp, which I hadn't looked at until I had already sent it, but it's May 27th, 2012 at nearly midnight Greenwich Mean Time. And in that year, July 4th, 2012, we announced the discovery of the Higgs boson. And this is one of the events that we took that's consistent with one of the decay channels of this Higgs boson, decaying into a pair of Z bosons, one decaying into a muon and an anti-muon pair, which you see as the red traces, and then an electron and positron pair you see as the green towers on either side of the experiment. And then in the middle you see kind of this little fireball of quarks coming out from the remnants of the proton collisions. And this is a visual image that representing all the ones and zeros and things that we measure that come out of that extremely complicated machine on the previous slide, and put into a visual form that we can recognize. We can see what's going on and show it to people who aren't scientists. Once we explain kind of the legend of what's inside of this picture. But there's a beauty to it, which I think is obvious to anybody. You see something there. You can make sense of different parts of what's happening in the form of this explosion. It's not chaos. But quantum mechanics is is a random. It's a part of the universe that's random in a sense. We don't know exactly what's going to happen in that particular collision. We could just predict the probability of what will happen, but we don't know what's going to happen. We can't force this collision to produce a Higgs boson. We just have to design the experiment and run it long enough to produce enough of them to get enough pictures like this to build up a statistical profile of the Higgs boson. But for me, seeing these types of the result of that means when I see that up there, it means my detector works, which is like the least trivial thing that you could possibly say. And because it mostly doesn't work, and and it takes years to design these things and operate them, and that so that timestamp up there tells me somebody was on shift at midnight in the control room taking this data. So we run this thing when it's running. It's 24-hour operation. It's always manned. So they're not down there, with the beams. You would die from the radiation, but they're in the control room operating the experiment. So we're able to do this kind of work only because of the friendship of our fellow scientists cooperating together peacefully in this joint pursuit of something that's much, much bigger than us. And I find that to be a very beautiful aspect of the kind of science that we do. It's not a solo endeavor. We really do rely on our friends to be able to do this. So it's through friendship that we've been able to discover the beauty of God, in a sense, which we could not have otherwise been able to do. And on July 4th, 2012, when we announced the discovery of the Higgs boson, the CERN auditorium, which is this big auditorium, was packed full of all the scientists, and we had we've had first our spokesperson present our results that showed we have very high confidence level that we've discovered this new Higgs boson in these various channels, and then after the next hour, the speaker for our competing experiment. So we have two big experiments where we're competing like two football teams. We don't share each other's secrets. We both have kind of the same rules, and then we use it as a way to cross-check each other, and also that competition encourages excellence in a way. But it gives you the ability to cross-check each other's results. So if you both have something that's consistent, that gives you a pretty good feeling that what you've discovered is probably correct. If one is right, if one has a result that's not consistent, something's not quite right, and that allows us to try to figure out what the truth might actually be. And so when I saw Fabiola Gianotti, our competing spokesperson's result that was consistent with ours, like this wave just went through the whole crowd. And people were crying, we discovered the Higgs boson. We've done something that started in 1967 and took us all those years of work. Like, it wasn't through lack of trying that it took us those 50 years to do this. And we did it, and we were right there together. So that was like, now, you can't, I probably wouldn't be able to repeat such an experience in my life. Yes. Right, but to have been there and to be part of that and to know that that's how we did it, I think that's another form of beauty for me. Yeah.
Just as a curiosity, how many people, more or less, worked on something like this? Like, are we talking about
thousands? It's thousands. It's like teams of 3,000 each, plus all the technicians and all of the staff and all of the support people, and they come from literally everywhere. When you're down in that cavern with this gigantic detector, you hear practically all the languages. I mean, the operating language of the physicist is English. The operating language of all the technicians and engineers is French. But then everybody else is speaking Italian. You've got Russians, you've got Bulgarians, Polish pipe fitters. It's just this and we're all down here in this cramped space with safety equipment on. It's just, it's like a science fiction movie, but it's real.
It's fantastic.
Yeah.
Okay, Massimo, can I proceed with the slide? You have slides, right?
Yeah, so let's I picked up a movie to show sort of a path here. It's a few seconds, but the idea is to start from something that looks pleasing to the eye in some way, more art form, we would say, which is a beautiful image taken with the Hubble. This was taken, one of the first one taken once we put the last instrument with the last shuttle servicing mission, and we had for the first time a camera that was able to work for the ultraviolet to the near infrared. So broadband observations and the stars appear with all the colors and they are all messed up. And the image is actually beautiful. But then there is here a little transformation. Just moving them around, you see that order happens. So there is a secret way to look at the image, which is the way that reveals a more profound level of order is the right word, or cosmos, which is the Greek word for order. And this is where I think the second level of beauty happens. What really attracts us is the fact that reality hits us with something pleasing to the eyes, etc. But then we are able to understand better that under that first aspect there is an order, is the right word. So there is a logos. An arche and a logos. At the beginning is this order, which is the real texture of nature that we try to find, to understand. And each time we find it, we say that's beautiful. No, there is this is at that level that really we resonate, which is amazing, as is the other slide, that we can understand it. That's because then we really are part of that logos with our brain, and this is where things get really interesting. So let's see if we can start the movie. I don't know if from there. No, it's not this one. That's the previous one. Go back.
Go back. Okay, here.
And there is a little here, I don't know if the person at the...
No.
Maybe someone has to do it from here. Yeah. There is a little audio as well. I don't know if we can hear it, but you can read the text. So this is a globular cluster, which is one of the oldest objects we have around our galaxies. There are about 150 of them. And they are like 13 billion years old, 12 billion years. So not far from the origin of time. And these are...
Hubble Space Telescope image of the crowded core of the globular cluster Omega Centauri. The brilliant colors of the stars are real. They correspond to stellar temperatures. They can also be used to trace stellar evolution. Astronomers like to know how blue the blue stars are and how red the red stars are. So we'll first sort these stars out by color, blue on the left and red on the right. Next, we'll sort the stars according to brightness, the brightest stars at the top and the faint stars at the bottom. The final plot you see represents different stages of evolution of stars. Stars spend most of their lifetime burning on the main sequence. When the fuel starts to run out, they expand to become red giants. They find a new source of fuel, helium, and burn blue hot. But even that runs out. They end up burning out as white dwarfs. Based on images for the Hubble Space Telescope, we've assembled a true plot that is a snapshot of the life history of stars in this ancient cluster.
It's like the next level. You see something, and we see this all the time. The images that we take with the Hubble, with Webb, etc., they are really pleasing, colorful, spectacular, and you see symmetry, But then what is interesting is that we can dig in and find out what is behind that. And this is the way nature works. And this is the point where beauty resonates with the ultimate structure of reality, which is a cosmos. And is a cosmos we can understand. And the next slide basically shows this type of path, because it's amazing that this type of plot, that one comes from a science paper, is sort of the same stuff. But no, with a couple of cartoons, you put the physics together, you put the equations, and then we can reproduce exactly that type of plot, starting with the math that we have invented. So this order can be understood. We do this all the time. There is a nature there. You must look at nature in the right way. You must reorganize things, and then our brain, our reason, understand it. And this is an interesting point, I think, for me of nature, which is of beauty, which is to understand this type of deeper level of truth that the reality offers us. So it's beyond the aesthetic level. It's really something that we are part of the same reality, of the same cosmos, because we are dust, stellar dust, basically, and we are able to understand how things are made, how we are made, and ask the next question: why? Know where we come from? Why is this way? And this is where I find, for me, the most profound and most spectacular level of beauty in reality.